Welcome to my channel, and today we will discussing purine nucleotide synthesis. This is the first step of our synthesis of purine nucleotides where ribose 5-phosphate converts into PRPP, which stands for 5-phosphoribosyl 1-pyrophosphate under the enzyme PRPP synthetase. ATP is being utilized here, magnesium is required for the enzyme, whereas we have inhibitors, purine ribonucleotides, and activator, inorganic phosphate. It's very much obvious here why purine ribonucleotides are the inhibitors here. Like for the process, the end products are going to be purine nucleotides, and when they are in an abundance in our body, obviously we will not require any more. So these nucleotides will act as inhibitors for PRPP synthetase enzyme to stop producing more PRPP, which is then going to push up the entire pathway into the synthesis of purine nucleotides. A very pretty first reaction. Going to be the second step where PRPP is acted upon by GPAT enzyme. This is GPAT. I have made a simpler abbreviation for the enzyme. The whole enzyme name is glutamine phosphoribosyl pyrophosphate amylotransferase, a very difficult name. So let's just go with the name GPAT. So PRPP is converted into 5-phosphoribosyl amine under the enzyme GPAT. Glutamine is used here. It is then converted into glutamate and excreted out. The inhibitors here are AMP and GMP. The activator is PRPP. The activator process of PRPP is very simple. Like obviously when PRPP will be present in excess in the body, it will push the reaction to move towards the synthesis of purine nucleotides, because PRPP doesn't just want to sit there idly. It wants to be in a reaction. It wants to be productive. Whereas inhibitors are AMP and GMP. AMP and GMP are the purine nucleotides, and these are the end products of the entire pathway. And obviously when they will be in excess, they will inhibit the entire process. Like the body doesn't want to have so much purines, obviously. And yes, these two points were also discussed in the purine salvage pathway. If you want to watch a video on purine salvage pathway, then you can look up in my channel. Anyways, we have 5-phosphoribosyl amine, which is then acted upon by GAR synthetase to form GAR. GAR stands for glycinamide ribonucleotide, a very difficult name. So I suggest you to go by the word GAR, which is simple. Glycine is used here, another ATP. GAR is then acted upon by GAR formyl transferase to form FGAR, and then formyl tetrahydrofolate is being used here. FGAR is then acted upon by FGAM synthetase to form FGAM, and another glutamine is being used here, and here is an ATP. FGAM is then converted into air by air synthetase, another ATP. Wow. Okay, so air then acts Okay, so air is now acted upon by air carboxylase and is converted into KRC and a carbon dioxide. It's easy. Then Kr is acted upon by SiCar synthetase to form SiCar. SiCar is then converted into ICAR by adenylosuccinate lyase, and ICAR is then into FICAR by ICAR formyl transferase again, and 10-formyl tetrahydrofolate will be utilized here. FICAR is then converted into IMP by cyclohydrolase enzyme. IMP cyclohydrolase enzyme. So till now we have got IMP. These are the further reactions of IMP. It has two options here. It can either go into the entire process to form AMP or into the entire process to form GMP, both of which are purine nucleotides. So let's see AMP first. IMP is acted upon by adenylosuccinate synthetase enzyme, and aspartate and GTP are being used here. Adenylosuccinate forms adenylosuccinate lyase, acts on it to form AMP. A fumarate is discarded here. And IMP is acted upon by IMP dehydrogenase to form xanthocene monophosphate, which is then acted upon by GMP synthetase to form GMP. A glutamine is being used here, and in this process the IMP dehydrogenase enzyme forms an NADH. Okay, so now we have got the final products, AMP and GMP. This is pretty much about the purine nucleotide synthesis. Now let's have a quick look at the inhibitors of the process. Well, we have PABA analogues, which are basically sulfonamides. These are the structural analogues of PABA, para-aminobenzoic acid. This PABA inhibits the bacterial synthesis of folic acid. In bacteria, folic acid is being synthesized in the body. So, and we know that folic acid is required here twice, this way. So if there will be no folic acid, there will be no purine synthesis, and obviously the bacteria will not be able to generate any kind of their DNA, their RNA, and this will kind of kill them. Whereas these drugs do not affect humans in any point, because humans do not generate folic acid in their body. They require external sources of folic acid. So these drugs do not harm humans. Whereas folic acid analogues, they actually inhibit this entire process in humans. These drugs include methotrexate and some related compounds, which inhibit the reduction of dihydrofolate to tetrahydrofolate, catalyzed by the enzyme dihydrofolate reductase. These drugs limit the amount of tetrahydrofolate available for use in purine synthesis and does slow down the entire DNA replication in mammalian cells. But like we can use these folic acid analogues for the control of cancer cells, but the disadvantage here is that these drugs affect, or I can say harm, all of the cells of the body. So they are like not good for all of the cells. They don't act specifically just on the cancer cells. Also, let's have a look at the reaction of folic acid being discussed here. I have tried to write down the whole of the reaction for you people. Dihydrofolate gets converted into tetrahydrofolate in the presence of enzyme dihydrofolate reductase. This is a reduction reaction in which NADPH is being used. So our drugs methotrexate, drugs like methotrexate inhibit this process, and though folic acid will not be available for the use in purine synthesis. Another thing here is mycophenolic acid, which is a reversible inhibitor of inosine monophosphate dehydrogenase enzyme. This drug, when inhibits this enzyme, it deprives rapidly proliferative T and B cells of the key components of nucleic acids. Obviously, AMP and GMP have to form nucleic acids like DNA and RNA. So if the cells are deprived of their nucleic acids, they are going to die anyway. And this is why mycophenolic acid can be used as an immunosuppressant to prevent graft reactions. This was all about our purine synthesis. I hope you liked the video and it was helpful for you. Leave down any suggestions if you have. Take care. Bye.